Good day, everyone, and welcome to KeyGene's two-part webinar on PanGenomes, the need and perspective. My name is Sheetal Dixit. I'm Director of Business Development at KeyGene, and I will be your moderator for today and also for the part 2 of the webinar, which will be held on March 18. During this 1-hour webinar, we will be having 3 presentations, and these presentations will focus on the why and the needs of the PanGenome and the value they can add to your breeding and your research. On March 18, we will be focusing more on the construction of the PanGenomes, so more of the bioinformatics part of it. At the same time, we will be also discussing the applications of PanGenomes in plant breeding and research. It's my pleasure to let you know that we are joined by 350 people who actually registered for this webinar. So thank you again, everyone, for that. We are happy and also very proud to have our guest speaker, Professor Rod Wing. Professor Rod Wing, he is the director of at the Center of Desert Agriculture and the professor of King Abdullah University of Science and Technology, KAUST. With me joining the other 2 speakers and my colleagues, Dr. Alexander Wittenberg and Dr. Erwin Datema, who are joining me live here from KeyGene Studio. And we are also joined by my other colleague, Eric Toussaint, who is actually at the backstage, and he will be helping us to get your questions to the speakers. So I'll request you all to type in your questions in the chat box, and we'll be happy to take them at the end of the webinar. So again, welcome everyone, and I am happy to introduce to our first speaker, Alexander. Alexander is the genomic scientist at KeyGene. He's considered as an expert in the next generation sequencing platforms, its use and unique applications. Alexander at KeyGene is also involved in many research projects with our partners and with many of the KeyGene successes in the next generation sequencing. Today, as a kickoff of our 2-part webinar, Alexander will tell us more about the importance of the high-quality PanGenomes, high-quality genomes, and its importance for PanGenome as a basis for a useful PanGenome construction. So, welcome, Alexander.
Thank you, Sheetal, for the kind introduction. Thank you. So, I'll start my presentation. So KeyGene, for the ones who don't know, KeyGene is the go-to ag biotech company for higher crop yield and quality. So we work in 6 different crop innovation platforms, as we call them. You can see them on the right. During this webinar, we will focus on the Genome Insights crop innovation platform. Within Genome Insights, we focus on a lot of different things, but I categorize them here in reference genomes and PanGenomes, which will be the main focus of today's webinar. But we also work on targeted sequencing approaches, genotyping approaches, transcriptomics, as well as data analysis and visualization. And the insights we gain within this Genome Insight crop innovation platform, they feed into the other crop innovation platforms. We use that knowledge, let's say, which we reveal in the Genome Insight platform in the other platforms like trait discovery and genomic and precision breeding. So we do many things in these. Today's webinar will focus on what I said on the reference genomes. So first of all, what is needed to generate such a reference genome? So high molecular weight DNA isolation and long-read DNA sequencing. And then analyze these reference genomes to generate a de novo reference. And then subsequently use these de novo references for PanGenome analysis, which Erwin will talk more in depth about. So going to the long-read sequencing platforms. So within KeyGene, we have access. We both use the Oxford Nanopore platforms. I'll focus here on the PromethION platform. Also the Pacific Biosciences Sequel 1 and Sequel 2 system we use in our research. So what does differentiate these 2 platforms? So I made a kind of a table here with the key characteristics, I would say, related to generating high-quality reference genomes. Because obviously with these platforms you can do more things like, for example, transcriptome sequencing, or amplicon sequencing, etc. But these statistics I show here are really focusing on generating long reads, let's say. So first of all, the differentiation can be in number of reads. So the Promethean platform, you can see the stats in the table, so I won't go through them one by one. Another key attribute is the read length. With ONT, you can achieve longer read lengths in general compared to the PacBio Hi-Fi approaches. The output is a differentiator between these two platforms. And with the PacBio platform, and in case you use the Hi-Fi type of data, there's really differentiation, let's say, in the raw read data, which is really high, up to 500 gigabases per cell. But that is being collapsed, let's say, into the higher accurate Hi-Fi type of data towards between 20 and 30 gigabases per smart cell. Another differentiator is the accuracy, which is very important, of course, for de novo reference genome assemblies. With ONT, with both platforms, I would say they are high, but especially the Hi-Fi, the PacBio platform, has an advantage currently over ONT in terms of accuracy. That Hi-Fi accuracy is very high. Another differentiator, especially if you're talking about PanGenomes, in which you want to sequence, generate data, let's say, for a large number of crop genomes, this can be an important differentiator. So the speed, the ONT platform basically enables you to start and run and sequence multiple samples in parallel, while the Sequel 2 platform does this in a serial manner. So the time for data production is a bit longer there. Another differentiator, which I did not put on the slide, but it could be in case you're interested in base modifications. With both platforms, it is feasible, but ONT has a clear advantage here, I think, in terms of that comes along with the data and is rather straightforward to get out of the data. So which platform to choose? Maybe this question comes up. So that really depends, I would say, on the research question, on the crop, the complexity of the crop, and the research goal, why you are generating a reference genome. So what we do at KeyGene is we, together with the client, we advise an optimal approach, let's say, which platform to choose to generate a high-quality reference genome. So why would you like to have a reference genome in the first place, actually, that really represents your germplasm? Well, that is very important to have a high-quality reference genome of your own germplasm you're working with in your breeding programs, because that is, let's say, the basis for a lot of things, for example, molecular marker development, to discover genes, to do comparative genomics towards other germplasm or public data, as well as focusing on trying to unravel the traits and to improve them, but also references genomes, let's say they look beyond single nucleotide polymorphisms, which is mostly used with short read data. With long read data and assemblies, de novo assemblies, you can also really look at other types of variations like structural variation, copy number variance, or methylation, what I just mentioned before. So now with the advent of these long read sequencing platforms, this is really no longer time or cost prohibitive. It really becomes, let's say, feasible to do this also for crops which are, let's say, not the high staple crops, but more the orphan crops or crops which, limited resources are available. So crop genomes obviously have a true challenge compared to, for example, diploid human genomes, in the sense that they have a very wide range of genome size. So they can range between 100 million base pairs for a genome like Arabidopsis, towards 150 gigabases for a certain flower type, but also many cereals and, for example, onion have very large and complex genomes. So what makes these genomes so complex? So first of all, many of the plant genomes have high number of repeats and also complex repeats, so very long and complex ones. Plant genomes can be heterozygous. And in case you want to face, let's say, these two haplotypes is a challenge, let's say. Some plant genomes can be polyploid, and for some species, plant species, there's a huge variation present, let's say, within the species. And you're only really good to, let's say, visualize and understand all this genetic variation when you generate reference genomes for the representative, let's say, species in such a variety. So genome sequencing, it's really about the 4 C's, as we call them. So it's about correctness, so basically the accuracy of the consensus sequence. About completeness, you would like to have the entire, let's say, sequence in your de novo assembly from telomere to telomere, ideally at a chromosome level. So the contiguity and the cost is obviously also very important. But now with these higher throughputs, the costs go down more and more. So these long reads, what I already mentioned, actually they can overcome these challenges of these complex crop genomes. Why? Because these long reads, they can span reads, can span the repeats, so they can be mapped more uniquely, let's say, on a certain place. And long reads you can, besides de novo genome sequencing, you can also use for transcriptome sequencing or identification of structural variants or DNA modifications, which I already mentioned. So to go back at the start, so what is needed for generating such high-quality reference genomes? Really, the first step is actually growing the material, I would say. So at KeyGene, we have over 25 years of experience in a wide range of crops for extracting high molecular weight DNA. We have developed direct tissue as well as nuclear isolation protocols for over 100 species. And last year we started a collaboration with Circulomics on also using their methods and to extract ultra-high molecular weight DNA in order to get even longer read lengths, basically. And currently this collaboration is still ongoing. A couple of examples here from both platforms, actually. So from the PromethION as well as the Sequel II. So here's melon. We do a lot of research on melon. Actually, here I compared both methods, so HiFi as well as the Oxford Nanopore platform, using their latest R10.3 pore type. And the table shows the main assembly specs and the BUSCO scores, so the gene completeness scores. We have many data about this. It's not all in the table, but it's just the key statistics here. We compared them. You can see that on the right bottom by doing an alignment and a dot plot. You see that on a structural level, the HiFi compared to the Oxford Nanopore the concordance is very high. And data is not shown here in the table, but when we look at the accuracy, let's say, especially when we use this R10.3 pore, it comes closer and closer to the HiFi data. It's not there yet, but so this is the way we, let's say, evaluate these two technologies to see where they are at and how they can be best used, let's say, for which crops. Here, another example in corn. We sequenced 3 lines, assembled them with quite impressive statistics, I think, good BUSCO scores. And at the end of last year, actually, there were some improvements again on the Oxford Nanopore flow cells, which has enabled us. We achieved around 165 gigabases from a single Promethean flow cell with an N50 read length of over 30 kb, which is really impressive. It represents about 70x, close to 70x coverage of the corn genome, which is, yeah, it's already sufficient to do a good de novo assembly. Here another example we performed for ELO Life Systems. It's a vanilla genome, planifolia. It was published beginning of this year in Nature Food. There we sequenced on the Oxford Nanopore platform, also generated Illumina and Hi-C data for scaffolding and facing, also performed the genome annotation. And actually in this project we were able to face both haplotypes, as you can see in the table below. And this reference genome enabled, let's say, the client to identify the vanilla genes in a better way, using these 2 haplotypes, and also elucidate in a better way the biosynthetic pathway of the vanilla genes and other aromatic compounds. So having this reference genome really enables, let's say, to do follow-up research which help the grower, let's say, to, for example, to work on all kinds of traits to in the end help the grower and breed for better varieties. Besides plants, we also work on pathogens, for example, plant pathogens. So this is a project we recently performed for the University of Amsterdam where we sequenced 96 Xanthomonas genomes on a single Promethean flow cell using native barcoding. Also here the yield was quite impressive, 125 gigs, with a N50 read size length of 33 KB. And we expect that this really will result. Research is still ongoing, but we think this enables, let's say, chromosome level assemblies with a high accuracy, and will enable, let's say, to better understand plant pathogen interactions. So moving to a few examples from the other long-read sequencing platform from PacBio, the Sequel 2. So here we have FabaBean. It's the one we sequenced was a diploid and homozygous line. It's a rather large genome and also complex genome. It's 13 gigabases in size, the estimated genome size. So we sequenced that using HiFi data and performed the de novo assembly using HiFiasm. Actually, we close to 12 gigabases total assembly size, and the Contig N50 was close to 3 million base pairs, and the BUSCO completeness here was 97%. This project is still ongoing, so the scaffolding is currently underway using Hi-C as well as bio-nano optical mapping, and all these efforts are being done in collaboration with the consortium effort, which is led by the Aarhus University in Denmark. More information you can find on this web link. So I already mentioned what is needed, let's say. So if you want to go to a reference to a chromosome scale, let's say level de novo assembly, in case you don't get there with just the long-read sequencing, there's other alternatives, let's say, to scaffold towards a chromosome level assembly. So we offer reference-based scaffolding using tools like RegTech, for example. We do a lot of bio-nano as well, optical mapping. We can integrate the contigs, let's say, from a de novo assembly using sequence-based genetic maps. Hi-C we use. POR-C we have currently in development within KeyGene's, so basically that's an alternative to Hi-C, but then using long-read technologies, so it provides even more richer data. So that is currently still in progress. So that brings me basically to the data analysis tools. So what we use a lot of public tools in general, both in the assembly field as well as in the consensus calling tools. But what we also do is we optimize these tools. We tune the parameters basically to find the best parameters for the assembly. And sometimes we also optimize them, for example, for speed, especially in case of large and complex genomes. Sometimes speed is important, let's say, especially if you want to analyze many of these genomes. And third, and maybe the most important, is that we have active development on development of our own algorithms, especially focusing on PanGenome reconstruction. And that is, of course, where this webinar is about. And Erwin will tell much more about all these things in the next presentation. So I think this is almost my last slide. So PacBio is a great technology, but we also think Oxford Nanopore is a great technology, and the developments with Oxford Nanopore are still going very fast. They have come a long way, and their read length are really amazing, and the read length of Oxford Nanopore really outperforms the read length of the PacBio platform. But accuracy is also very important, and ONT has made tremendous progress, let's say, on that topic over the years, and is still making progress on that. So currently we're in the so-called Q20 chemistry early access program. I think or hope the cells are being started today, let's say, for that new chemistry. So that really, the aim there is to even increase the accuracy even further, so that it comes on par with the HiFi assembly accuracy. So, but that is all work in progress, but I just wanted to give you a small glimpse of that. So basically, to summarize, there's really three important things for a de novo assembly. First of all, is good starting material and expertise in ultra-high molecular weight DNA isolation. Of course, then you need to have the expertise on the long-read sequencing platforms. Finally, state-of-the-art data analysis, integration, and visualization tools are, let's say, key to first of all obtain good references, but also, let's say, to make use of these references in terms of in relation to other data types and in terms of use, let's say for the end user, for example, the molecular breeder. So then visualization tools come into place. This is my final slide. So this shows, let's actually say, a step to the PanGenome area. A single reference genome doesn't tell the entire story. Really, natural variation is a source for new traits, and that's why it's so important, let's say, to not, let's say, restrict yourself to a single reference genome, but look further into the diversity within the species to generate multiple reference genomes and see how the relationship between these genomes is to get really the most out of your data. Thank you for your attention.
Thank you very much, Alexander. That was really very interesting, and thanks for sharing the insight. You really set the stage, I would say, for this topic, because you reinstate the importance of high-quality reference genome, because garbage in is garbage out. So before we talk about generating high-quality PanGenomes, these are the foundational things that are important to keep in mind. So thank you again. If you have questions for Alexander, this is the time now to tap into the chat box. So this gives me a moment to introduce to our next speaker, Dr. Erwin Datema. Erwin is a senior scientist of genome informatics at KeyGene, and Erwin will show us today the why of the PanGenome. And also Erwin will show us a glimpse of KeyGene's capabilities into building PanGenomes. Erwin, welcome.
Thanks for your introduction, Sheetal. So today I would like to talk about how we are unraveling genomes at an unprecedented scale and how we use these genomes to construct large-scale PanGenomes. So, as you know, with the technologies like the ONT Promethean or the PacBio sequel, we can now sequence many genomes, and not only single reference genomes, as Alexander explained, but also multiple genomes of the same species. So let's say a cherry tomato and a beef tomato and a round tomato. And of course, we want to compare all these genomes to each other. And in that sense, we are entering the PanGenome era. So why are we doing this? The most important realization that we have had over the past decade or so is that these genomes, especially in plants, vary tremendously from each other. So, for example, in Brassica, about 10% of the genes are only found in a subset of accessions. And this basically means that every genome is unique and that your germplasm contains many unique traits that are not present in other germplasm. And so in that sense, it is important to start sequencing these genomes and start discovering all this variation. And that not only goes for Brassica, of course, but also for other genomes like maize, where it was found that about 25% of the loci could not be anchored to the reference genome that is present. And many of these loci are enriched for important agronomic traits. The same story with rice. And of course, we have examples of many more crops. These are just a few I would like to highlight from the public domain. But what this now means is that we want to look at this genome in a different way than we have done before. So, for example, around 2012, we at KeyGene, but also many other people, were looking at genomes in a form of what we call multivariome analysis. So, starting from a reference genome that often consists of chromosome level scaffolds or maybe even smaller contigs, we start aligning these short reads. So from a different variety. And then we can align the short reads from another variety and identify polymorphisms between these varieties. So what we call SNPs, simple sequence variants. But also we can start looking at gene copy number variations. And this allowed us to, let's say, unravel already a lot of differences between the different genomes that we were looking at. But as time progressed, we also got different ideas how we could look at these genomes. So now starting again from a single reference genome, we would again map these reads to that genome, but also take a look at the unmapped reads, so the ones that we could not place on the reference genome, because we were getting to this insight that there may be a lot of interesting information in there. And by assembling these unmapped reads de novo, we could then add them to a kind of extended PanGenome, so a kind of linear representation of the entire genome space of these different species, and then align the second variety to it, and continue the same approach by taking the unaligned reads, assembling them de novo, and adding them to the PanGenome. And in that way, we were able to discover many unique genes that are present in only a subset of the varieties that we're looking at. Of course, the story doesn't end here, because now with this long-read technology, we are able to sequence entire genomes de novo, and we also need new ways to analyze and compare these genomes. So over the last couple of years, we have been working on this PanGenome graph approach, where we take a de novo assembled genome from this long-read sequencing technology and then compare it to another de novo assembled genome by just performing a genome alignment. And of course, you can extend this idea to having multiple genomes present. And now you can identify the regions in these genomes that are similar between subsets of those genomes, or maybe unique to one or two of the genomes, and in that sense identify those regions that could be of agronomic interest. So each genome now is a kind of path through this graph. So not every block is present in every genome, every block of sequence, and some may follow another order than another genome. And in that sense, we can now compare these de novo assembled genomes to each other and identify, let's say, the shared and unique gene content, but also the structural variation, simply based on how these genomes align to each other. And of course, this has a lot of added value. So if we now start from these genome insights that we get from the multivariome approach, we kind of do a biased all versus reference approach. So we can only identify variation in what is present in the reference genome. Whereas with this second approach, this linear pangenome approach, we could also start looking at the pieces that were not present in the reference genome and identify variation there. But of course, it's also crucially important if you want to compare genomes and make crosses between individual plants, that you know the structure of these genomes. So to go from these genome insights to the precision breeding, we first started with nucleotide variation and gene copy number variations at our disposal. But as the technology matured, we also could look at novel genes. And finally, with this novel graph-based approach, we can now also look at the comprehensive structural variance between all these genomes and know how the genomes are organized with respect to each other. And so that brings me to what we can do with this. So if we are looking at multiple genomes at the same time, we can also start looking at haplotypes, and we can start looking at haplotype patterns. So for example, you take two genomes and make a cross between them, then all the offspring will share parts of the genome from their parents. And by making an inventory of these, you can easily identify those regions in the genome that are unique to a certain trait or a certain variety that you have. And that allows you to now identify these haplotype patterns and improve your breeding program in that direction. And of course, that doesn't only go for biparental crosses. This also works if you have a breeding population. So once you construct this pangenome, you can then identify all these haplotype patterns that are available in your germplasm and identify which pieces of each haplotype, which pieces of each genome are responsible for certain traits or phenotypes. Now, in order to do all this, of course, we need a comprehensive computational tool set. Many of these tasks are very challenging, and Alexander already told us about many tools that we use from the public domain. But not only that, we are, of course, also developing our own tools, because many tools that we use are developed mainly with a focus on human or diploid genetics, whereas we work with many crop species that have much more complex genetics. And so in order to analyze these and build pangenomes from this, we started building our own tool set. And so just to give you a little taste of this, in my next presentation, I will tell you much more about it. But what I want to show now is that, for example, when we take 3 rice genomes that have been sequenced in the public domain about 10 years ago, making a pangenome of these with public tools like seq-seq-pan or progressive MALV takes a couple of hours. Whereas our own technology that we call STL allows us to compare these 3 genomes in a minute. And of course, this scales very well to also larger genomes. So for example, if we now take a single chromosome from a large genome that is about 100 MB, the chromosome, we take 9 accessions of this and try to build a pangenome out of that with the public tools, well, seq-seq-pan did not finish within a week. Progressive MALV took about 100 hours or 4 days. Our own tool, STL, allows us to make this comparison in 2 minutes. And that really gives you, let's say, power that you did not have before, because you do not have to wait anymore for these results. You can immediately start discovering all the variation in your genome. And of course, this doesn't end with single chromosomes. This scales to large polyploid genomes even, where the tools that we can find in the public domain do not even finish within a week. And we can build a pangenome of 9 polyploid genomes that are about 4 gigabases in size in an hour. And these are some of the examples that I show today. In my next webinar next month, I will show many more examples where we go even bigger and faster. And so with that, I would like to end the presentation.
Well, thank you very much, Erwin, for your presentation. For me personally, it was very deep insight, but presented in such a simplistic way and explaining the whys of the PanGenome. So thank you. If you have questions for Erwin, please type your questions in the chat box now. And please remember, Erwin will join us also for the part 2 of this webinar series on March 18, where Erwin will dig more deeper into the construction of the PanGenome. And he will also share some of the case studies or the success stories, showcases, if you will, in his presentation on March 18. So if you have not registered yet for that day, and if you're interested, please do so now. So thank you again, Erwin. And it's now time for me to introduce to you to our final speaker, and that's Professor Rod Wing. Professor Rod Wing is a renowned plant scientist who has taken professorship positions in prestigious universities like University of Arizona, Clemson University, and Texas A&M. Currently, Professor Wing is the director of Center for Desert Agriculture and a professor at King Abdullah University of Science and Technology. His lab is interested in the evolutionary history of rice and focuses on detecting natural variations by generating high-quality reference genomes for cultivated rice and its wild relatives, with the aim of creating a pangenome. Today, Rod will tell us about his views and experiences in the needs for a pangenome and the value of pangenomes. Welcome, Rod.
Okay, hi. It's nice to see you all. I'm calling in from Arizona, and thank you very much for the invitation to speak. Can you hear me okay? Everything's good?
Perfect, Rod, go ahead.
Okay, excellent. Okay, so let me get my slides going here. Okay, so this is the beginning of my talk. And let me just get adjusted here with my video. This one, I'm sorry.
No worries.
All right, can we make this smaller? A little bit smaller. I can't see my slides there. Okay, that's better. All right, I'm in good shape now. All right. So today I'm going to talk about we call this the Pan-Ariza project, harvesting 15 million years of Ariza evolutionary history to help solve the 10 billion people question. And it's exciting to be at this webinar. I've interacted with KeyGene for many, many years, and that's why I wanted to bring up this first slide. My first seminar at KeyGene, I'm going to say it was about 20 years ago. They gave me this puzzle game called Frag Mind, and this is you get a card and you line up all the pegs together, and then you have to align them into an alignment, and so all the colors align properly. So all the greens here and things like this. So. I haven't played a lot of this game before because I'm colorblind. It's hard for me to do the reds and the greens, but once somebody sets it up for me, then I can figure it out. But this kind of is I'm starting to think about, 20 years ago, we were thinking about, really, PanGenomes and so on and so forth. So you might want to play this game to, at some point. It's pretty cool. So before I get started, I want to just tell you a couple things, a couple of the projects we're working on at KAUST as well as the University of Arizona. I still have my wet lab operating in Arizona. So our main project that we've been working on for many years, since 2003, is called the International Rhizomap Alignment Project. And this is a comparative evolutionary genomics project for the entire genus Ariza, to study its evolutionary history and to use the information to improve cultivated rice. We've moved now into some projects related to the neo-domestication of some of the wild relatives of rice, and I'll mention that at the end of my talk. a big goal of these projects is to essentially create a digital gene bank of crop plants and their wild relatives. And we're working on rice right now, but we'd like to extend that to other crops. And lastly, I've launched a project called the KSA Native Genome Project. And this is to try to generate high quality reference genomes for all terrestrial and aquatic life forms in the KSA for environmental protection and monitoring and preservation of biodiversity. So we definitely need to develop many genomes. And now with the kind of the PacBio, the Oxford Nanopore technologies, this is becoming a routine. Okay, so one of the kind of the driving force that keeps that I kind of wake up to every morning to kind of keep me thinking about the work we're working on is really the 10 billion people question. How do we grow enough food to feed the world in less than 40 years? And rice is going to play a central role in that project, in that helping to solve that problem. As it feeds half the world, it's a rice-dependent population that will double by 2050. So a big goal now is to develop new varieties that are much higher yielding, are more nutritious, but also have less of an environmental footprint, can use less water, fertilizer, pesticides, and can grow on marginal lands. In some circles, this would be called green super rice. So how are we doing this? We're actually trying to understand natural variation across rice and its wild relatives. And this slide demonstrates that you can find diversity in rice for almost any conceivable trait. This shows, like different seed colors, seed size, seed shape, panicle architecture, and plant architecture. These are just, kind of naked eye polymorphisms, but there's many, many more that are buried inside the genome. So the genus Oryza is shown here. This is a phylogenetic tree. We have 27 species, wide distribution in habitats, 11 distinct genome types from the AA to the GG, and there's many polyploids. And there's also a 3.6 genome size variation. So this is the wide distribution. This is where you can find the wild relatives of rice. And this is the genome size distribution right here, from about 360 up to 1.2 gigs. The wild relatives of rice, of course, are also phenotypically diverse. And this just shows an example here. Here are the two cultivated species, Oryza sativa, which is what we all eat, Oryza glaberrima, this is African rice, and this is a progenitor of Asian rice. And you can see all the variation here. So even this looks almost like could be like a maize plant. Yeah. What we like to say is the wild relatives of rice are agronomically inferior grasses, but contain a virtually untapped reservoir of genes that could be used for crop improvement. So fortunately, the International Rice Research Institute in Los Baños, the Philippines, they have a gene bank that has about 130,000 accessions of rice, including the wild relatives. And I have a joint appointment there that was founded through the AXA chair. And we to get a early handle on the natural variation in rice, in cultivated rice, I was part of a project where we essentially sequenced 3,000 accessions of rice and mapped that data to Nippon-Bari and a few other reference genomes, and to learn about the natural variation. One of the conclusions that came out of this and further work down the line is that we could essentially subdivide these 3,000 accessions into 15 distinct subpopulations. So we have Nippon-Bari, of course, which is the original rice genome sequence that was sequenced many years ago, and it was published in 2005. very, very high quality reference genome. And so everybody's been essentially mapping all their data to Nippon-Bari. But when you look at the population structure, you can see here that there's a tremendous amount of variation that exists. And so we're trying to get a good handle on this problem. Mm-hmm. So one of the things we want to do with this 130,000 accession database is essentially generate what we call, again, a digital gene bank for cultivated rice. And a goal here is to generate ultra high quality reference genomes. We call these platinum standard reference sequences that represent the 15 subpopulations of Asian rice, resequence 10,000 rice successions at a high coverage, detect all SNP and structural variation relative to all 15 platinum standard reference sequences to create what we call a pan haplotype map of Asian rice, resequence the remaining collection at low coverage, and then impute structural variation and SNPs across the entire gene bank. And in parallel, develop mapping populations and use that to essentially assess, to phenotype the mapping populations as well as the many of the accessions using a global rice array. And this is a concept that was developed by the former Director General of IRRI to essentially phenotype, let's say, up to 10,000 diverse successions of rice in many locations around the world, and gather that data together along with genotype data so we can essentially breed for almost any trait at any time for any environmental condition. So one of the key principles that I have learned over the years, I mean, our lab was involved in developing the original rice genome sequences, that a key element to understanding, capturing, and exploiting natural variation across the horizon is to have access to ultra high quality reference genome sequences that are contiguous and gap-free, which means they're superior in quality across all regions, not just gene space, which I kind of call gas assemblies. And so we're calling these types of ultra high quality reference genomes as platinum standard reference genomes. So I'll take you back to 2005. This is a different representation of the 15 subpopulations. But back in 2005, we had a single reference genome, and this was a $200 million effort with 10 nations. We got this down to about 200 gaps. So the back-by-back approach, and all the backs were finished. And it took many years to develop ultra high quality reference genomes after this one, and this all has happened fairly recently with the advent of long read sequencing and the reduction in cost. So in 2016, there were essentially 5 to 6 ultra high quality reference genomes that had been developed for rice. Our group was involved in Mingwei 63 and Shenzhong 97 back in 2016. These were the first PacBio reference genomes for cultivated rice. This was led by Zhangwei Xiang, who's now at Huazhong Agricultural University in China. We did IR64 back in 2018, and N22, which is an Ausch drought-tolerant accession back in 2018. So since then, with the decreasing costs in PacBio, we've exclusively been using PacBio. It's kind of our go-to technology right now, and we've been very pleased with this technology. And it's and now with the hi-fi reads, it's incredibly easy to generate a high-quality reference genome very, very, very quickly. This is so we ended up making high molecular weight DNA for the remaining 12 accessions, and this was before hi-fi. So this is CRL reads, things like that. But we generated this amount data, this type of coverage, and these were our mean submarine lakes, and from let's say 11 up to 20 kbs in size. Then we ran Platinum Standard Reference Pipeline assembly results, where we essentially use 3 assemblers and then go to consult Bionano optical maps, and then we use a software program called Genome Puzzle Master to edit the contigs, and then we do quite a bit of polishing, and then generate the pseudomolecules, and then one final Bionano map validation step. These are the results for the remaining 12. We had between 3 and 92 gaps, and this has since been corrected, the IR64, and that was an earlier version, but this is now down to into the teens. This just shows an example of a Bionano map validation. So this is a completely independent validation of the assembly, and for chromosome 1, in this case, there's a single gap, and it's in a centromere repeat. So we completed this work back in December of 2019 and released this in bioRxiv, and this was published in Scientific Data in April of 2020. So all the genomes have been assembled and released along with all the data, and now a preliminary analysis of transposable element and presence absence variation across these 12 genomes, as well as the entire population structure of cultivated Asian rice. One thing I'd like to mention is at Christmas this year, in 2020, John Wei Shang and Ling Chen and Jesse Poland, we would collaborate on this, released Mingwei 63, new versions of Mingwei 63 and Chenchang 97 that are essentially gap-free rice genomes, end-to-end, and telomere to telomere for most of the chromosomes. So we're very excited about this. These were the first crop genomes to be released that were gap-free. So this is in public databases now for your analysis. So we have these genomes now. So our next process is really genome annotation. So we're collaborating with Doreen Ware's group at Cold Spring Harbor, and we've been longtime collaborators with them. We have panicle, leaf, and root tissue, RNA-seq, as well as iso-seq data, and going through this particular pipeline here for all 15 subpopulations plus Nippon-Barre as a apples to apples comparison, plus a number of other wild AA genomes. They're all being re-annotated again using an updated pipeline, and this data will all be available at the end of February. I'd also like to mention another project that we're working on with Andy Jones, who's the lead PI. This is a National Science Foundation BBSRC proposal called the PanHorizer Project. And essentially, this is to unify the gene calling nomenclature across the PanGenomes. Because right now, all the genes are completely different. It's really hard to find like one gene and say, well, okay, you start out with one gene. Well, what does this look like across like all 16 genomes, for example, and what's the variation, and so on and so forth. So this is really from a kind of a protein level. So we're working with EMBL and Uniprot and another group at US at Oregon State and Erie. And these are the goals right here. So I think you guys will have copies of these slides so you can read that. One thing I'd just like to mention is one thing that Young Zhao in my group has been doing is essentially comparing looking at inversions across all of these genomes. And we've identified genome-specific inversions, population-specific inversions, and it's looking like at least between the 16 Asian rice varieties, there's about, I'd say, somewhere about 30 to 50 megabases of sequence that's inverted relative to the Nippon-Bari genome across all these genomes, and we're basically characterizing these in great detail. With respect to the wild relatives of rice, so not only are we doing a pangenome of Asian rice, we're also doing a pangenome of the entire genus. Again, I'll remind you the phylogenetic tree. And one thing that we're focusing on right now, we've already done all the sequenced all the AA genomes and upgraded these guys with PacBio, and now we're focusing on the polyploids. So I'll just kind of let you know about 3 polyploids here. These are the CCDD genomes. These are all from South America and Central America. And in a collaboration with Jian Li's group in Beijing, China, we just released an assembly and a detailed functional analysis on entitled "A Route to De Novo Domestication of Wild Tetraploid Rice." And this is not only the assembly, but also kind of to neo-domesticate these guys. Because many of these species can grow in extreme environments, and so rather than teaching Asian rice how to grow in the extreme environment, you domesticate a rice relative that can already grow in those environmental conditions. Another set of examples here. This is the KKLLs or the HHJJs complex right here. We've been focusing on Oryza cortata. Oryza cortata is the only halophyte of Asian rice, of Oryza. It has a biological range from Myanmar to Pakistan. It can grow in brackish water. This is what it looks like. It's actually eaten in Bangladesh as a winter sweet rice, but it shatters and it has a number of properties. So we've developed a high quality genome assembly for this guy. This is the Bionano data, and we are now involved in annotation, functional genomics of salt tolerance, collecting voucher specimens for population genetic studies, and developing a transformation system to neo-domesticate Oryza cortata. Okay, so lastly, I think that's the end of my talk. I'll just say that I'm excited to be here. I want to learn more about how PacBio, not PacBio, how KeyGene is able to generate a pangenome in minutes, and we'll probably jump on that boat. I think I'm excited to learn more about that, and probably learn more about that before the next seminar. But the genus Oryza is a fantastic genus to work on. It represents 15 million years of evolutionary history, and you can look at various time frames when you want to study the evolution of genomes. You can look within 10,000 years within the cultivated species. You can look within 2 million years with the AA genomes, and then we can go all the way out to 15 million years. So there's so many basic research questions as well as applied research questions that could be addressed from this tremendous resource. And we expect to have the I think it's 42 Platinum Center reference genomes released and publicly available by the end of this year. With that, I'll just thank a number of my collaborators over the years, the IOMAP consortia, my lab in Arizona, former lab members, and my new lab in KAUST, which is essentially a computational genomics lab. And with that, I'll take questions. Thank you very much.
Thank you so much, Rod, for sharing this interesting work that your lab is doing. Really, I think this is a nice example of how innovation in technology can drive new applications, which I think we all are looking forward to. So thank you very much. I think this really marks for now opening up the discussion for the panel. So now is the time to take our audience questions. So I'll now give the mic to my colleague Eric, who will read out the questions from the audience for our speakers.
Hi, Sheetal. Yes, let's start with a question for our guest speaker, Rod. Manish asks, are you also searching for structural variations like translocations, duplications that are conserved across those 16 genotypes of rice? And if yes, so in fact 2 questions, then how do you define that a duplications is conserved in multiple genomes?
Rod, did you hear that? Could you hear that? No. Eric, can you repeat?
I couldn't hear anything.
Okay. Eric, Rod cannot hear your question. Yeah, that's
Perhaps you could repeat the question, Sheetal. So the first question is, are you also searching for structural variations like translocations and duplications that are conserved, so the same, across all the 16 rice genomes?
Yes, absolutely. The inversion one was just kind of a we began with that, but we didn't have all of our genomes annotated. But now that they're fully annotated, it's going to be easier to do that. But we're trying to study all structural variation.
And in fact, the second question of Manish is, now you're studying those, how do you define that the duplication is conserved in multiple genomes? Do you have parameters to say yes or no?
Well, yeah, I mean, if it's duplicated, then, we will confirm it, if it's in like a second genome or a third genome or a fourth genome, so on and so forth. I mean, that's how we that's the nice thing about having a PanGenome like this. You're able to track the history of the inversions, and then we have various outgroups, and we can go to the original, like Rufapogon or Navara, which are the putative progenitors of rice, and then we can go all the way to the B genome, which is punctata, which will take us, which is the outgroup for all the AA genomes, so on and so forth. So there's definitely parameters on this. I'd be happy to talk to you more about it.
All right. Eric, do we have more questions from the audience?
Yes, indeed we have.
Okay.
We have a question from Abner, and it's a bit again about parameters. How does one say when a reference genome has a high quality? When can you consider a reference genome having a high quality?
Is that for me?
Do you want to take this, one of you, Erwin?
Yeah, so I mean, a high quality is of course very relative to something else. So, but I think important parameters there are the number of gaps that you still have in your genome, the number of unresolved regions that you still have, and let's say the number of scaffolds in general. So ideally, a high quality genome would be telomere to telomere, and without any gaps in them, and without any sequence errors in them, and with all, let's say, variation represented. But yeah, that's very challenging.
If you don't know a ground truth, that's basically I think to add, then it's you have to compare it with something. But so that's what we can do when the technologies, let's say, iterate. We get a better feeling, and when we generate alternative types of data, like also Rod was showing the BioNano data, these type of alternative types of data can also validate the data you have generated with generating a reference genome. So there's all kinds of methods to do that.
Right. Yep.
We say our truth is the BioNano maps, because these are optical maps. These are basically like restriction maps of native DNA fragments. So and we try to make our assemblies match the bionano maps.
Right. Yeah. Well, thank you. Well, I see I have a question coming in. Rod, this is for you. How would you describe the main bottleneck in PanGenome approach? Is it the choice of the line that one needs to include, or is it data generation? Is it analysis? Is it visualization?
I would say now for us it's the analysis part and getting it right. I mean, it's what do you? It's kind of like what do you call a pangenome? I mean, I think we actually have with like these 16. We call it the magic 16. It's 15 plus Nippon-Bari. Right. I mean, that actually is a pangenome, but it's not a graph. And I don't know how useful the graphs are going to be, but it's and then it's getting associating the phenotype data with these genomes. So we have a project we've begun with Erie recently to do a drought study, and we've selected 15 representatives of each of the 15 populations, and we're doing a drought test on those and using basically resequencing these genomes with PacBio, so long read resequencing to get a handle on long-range structural variation as it relates to drought tolerance. I think that'll be a pretty interesting study to do. But it's really, I think it's the analysis and the phenotype data to generate a. Believe it or not, I mean, I'm an old guy, but my daughter and I sequenced asawe rice, which is a Saudi Arabian rice, in 2 weeks, under the watchful guidance of two very skilled technicians during the COVID break. We'll call it the COVID break, and it was exciting. It was just pretty fun to be in there, and we got a killer assembly, and it's really easy to do it now to generate at least the raw data for high quality genome, right. And then if you have the computational tools to assemble it, it goes like that.
Right. Yeah. Thank you, Rod. We have more questions for our speakers. Eric?
Yes, we have a question about the costs. Can one say something in general about how much a pangenome would cost when you want to generate and use it?
Maybe Rod can add, but Rod, it's definitely not anymore what Rod mentioned, what the first reference genome of rice had cost and what efforts went into that. All these were, let's say, consortium efforts, similar to the human genome. Which took years with many groups, and nowadays, you can the data generation at least is very straightforward. So the costs have come down tremendously, I would say. But always with cost, it's what are you talking about? What type of quality reference genome? What type of which crop anyway? What to include? Annotation? Yes or no? So cost is always relative, but definitely in at least in terms of data generation, also the data analysis, of course, these costs have come down tremendously. Also with the assembly, let's say previously with, for example, with PacBio, but with ONT, the same. Assemblies could take, even if you had enough sufficient infrastructure, assemblies could take weeks or months to finish. And now with the higher quality data, both from PacBio as well as from Oxford Nanopore, actually, let's say from a 1 gigabase genome, you can do a de novo assembly in a couple of hours, maybe even, or sometimes even less. So this all has brought down the cost further. Rod, maybe you can add further?
No, I agree with everything you said. I mean, it's I'm not going to give a quote, but I think it depends on smart cells. I think smart cells now, at least in the public domain, or somewhere to run a fully loaded smart cell somewhere around 3K. So if you need that's US dollars. If it depends on really how many cells you need, and then there's the important, again, one of the key things that everybody forgets, and KeyGene is right on top of this, is you need really high quality high molecular weight DNA to really make it happen. And our lab specializes in this as well as KeyGene. And there's a lot of groups out there that they're like one-offs. They want to get a genome, so they send their DNA into a company, and you can't sequence it because it's I think, as you said, Chital, like crap in, crap out type thing.
Right.
You really need professionals that know what the hell they're doing.
Right.
And because it's not trivial.
Absolutely.
Especially, indeed, that's a good point, indeed. Especially in case we scale up to PanGenomes, so you want to do hundreds or tens of genomes, and this becomes even more important. Also, with the different varieties, there comes different challenges in the extraction of the DNA because, these different varieties, they look so different. So also their metabolites and the structure of the tissues is also different, most likely. So, indeed, that's a very important point.
Yeah, and to add to that, I mean, over the last decade or so, there has been a tremendous drive to get to the $1,000 genome or the $100 genome. But I think what is getting more and more important is, can we do the $1,000 genome analysis? And as the costs for sequencing and data generation go down, we start to sequence more and more. But that also means that the cost for analysis goes up. Yep. And of course, also on that, we are working very hard to make sure that the $1,000 genome really becomes a $1,000 genome and not a $1,000 genome plus a $100,000 genome analysis.
Good one. Yeah. Thank you. So we have more questions coming in. Looking at the time, Eric?
Yeah, sure. Sure, we have some more questions. One for Erwin. Is STL Progressive already available, or will it be published scientifically?
So at the moment, our intention is to keep these algorithms in-house. We are still actively developing them. And of course, it is for us very interesting to come into contact with you and actually help you with your analysis.
Right.
Okay.
Mm-hmm. All right. Eric, do you have more questions?
Yes.
All right.
So, do we here in KeyGene? There's a question from a guest. I didn't get the name as yet. He or she is interested in the de novo reference genome that is telomere to telomere and gap-free, so a full chromosome. And are there any tips to accomplish this level of standards beyond which sequencing instrument and coverage? Yeah, I think it's for Alexander, isn't it?
No, I'm thinking it's a good question indeed. So it really depends on the genome structure. Let's say how complicated that is. So without knowing that information, or maybe it is not known at all, but without knowing that, it's difficult to comment on that. Besides the more trivial things which we already mentioned, that high molecular weight DNA isolation is very important, is key, independent of the platform, I would say. And what we have seen in the past, but what we would like to get rid of, basically, and we see that that is getting there already, is that we would like to move towards one technology approach only. So that is not necessary anymore to add additional data types because, first of all, that lowers the cost, and second of all, it also makes the analysis easier and more straightforward in case you could do that. And now, I definitely, I think with both technologies almost, and it's definitely moving there, that this becomes possible and feasible. But it is really dependent still on which genome you're looking at. A rice genome, which is homozygous, I suppose, for example, which is just an example, which is 300 megabases and homozygous diploid, that is easier to de novo assemble and sequence compared to a heterozygous 1.1 gigabase polyploid rice variety. So that is just more challenging. So it depends on the complexity of the genome you're looking at, basically.
Mm-hmm. All right, thank you. Eric, looking at the time, maybe there's room for one or maybe two questions, last questions, before we close the session.
Okay, Chital, I have a question which is about vision or predicting the future. Jan asks, how about trait mapping in PanGenomes? Will what we know as genetic maps still be useful in, say, a few years from now?
genetic map is a kind of low-resolution representation of your genome, or actually of 2 genomes of a population. So in a world where we can sequence these entire genomes and assemble them for a similar cost, no, there is no use for genetic maps anymore. But I don't think we are in that world yet. So I think for the next decade or so, genetic maps will still have a very specific value for those applications.
Okay. Well, okay. Well, Shita, I think we managed to cover almost all of the questions.
Very nice. All right, so thank you everyone for the questions, and I think that brings us to the end of the first part of the 2-part webinar series on PanGenomes, and I hope you all enjoyed this part. And of course, if you are interested to learn more about how in the construction of the PanGenomes, and to also understand how this can be applied in plant breeding and your research, please join us in the part 2, which will be held on March 18. So, and in the meanwhile, if you have questions, please do feel free to contact us on the link below, which is partnering@keygene.com, and also you can look it up on the website. Of course, if you have questions for our guest, please send them to us for Rod. And in a minute, right after this webinar, we will be sending you an invitation to review this webinar. So we would appreciate a lot if you can give us a feedback. This helps us to improve our webinars. So again, thanks a lot. And I would like, with this, to thank all our speakers, especially Professor Rod, who has been joining us live from Arizona. Thank you so much once again. And thank you, Alexander and Erwin, for this very nice interactive webinar. I hope you all enjoyed this and found it valuable. And I hope to see you in the second part of the webinar again on March 18th. So if you haven't registered yet, please do so. It's free, and we would like to see you there. I would like to remind you that on March 18, our guest speaker will be Dr. Alan Sessions from BASF, and he will be presenting his work at BASF on how they are applying PanGenomes in their current work. So stay tuned for that. For now, I want to wish you all the very best. Take care, stay safe, and I hope to see you in March. Thank you, everyone. Bye.
Bye.